Okay, so hello everybody. Thank you so much for joining us for today's SEDS Online webinar. My name is Chelsea Pedersen, and before we get started, I want to thank our sponsors, the IAS, which allows us to offer all of the resources free of charge to you. And so make sure and check out the website. It has lots of different types of learning tools, recorded lectures, virtual field trips, you name it, it's on there. So please take a look and yeah, let us know if there's any questions. And today's lecture is by Dr. Daniel Aristogui, who's a professor at the University of Geneva. He's also the president of our sponsor, the International Association of Sedimentologists. And he received his MSc from the University of La Plata in Argentina before heading over to Erasmus at the University of Miami as a visiting student. He then went to ETH Zurich for a PhD and a postdoc, and then went over to the University of Geneva. He's extremely active in the research community. I found out yesterday that he has over 300 research publications, really impressive. His focus really is on marine and lacustrine sediments as paleoenvironmental archives and indicators of anthropogenic impact on past and present environments. And he's recently been focusing on applying geomicrobial approaches to modern environments and trying to develop different proxies. So today he's going to talk to us about microbial environments from both laboratory and lacustrine settings. And with that, Daniel, thank you so much. We're excited to have you, and I will give you the mic.
Well, thank you very much, Chelsea, and thanks very much to SEDS Online for inviting me and for setting this wonderful series of talks and webinars that are really, really a wonderful thing for all the sedimentological community. And before going into the talk, I would like just to, I see that in the audience there is a lot of young people, so perhaps I need to give a little explanation about the title. And you know, back in the 60s, there were a series of movies that were called the spaghetti western films because they were most of them done in Italy and some of them in Spain. And one of the most famous one is this one, that is The Good, the Bad, and the Ugly, where three characters, very different characters that you can see for the names, were sort of somehow working towards a common goal, but in between them, there were a lot of problems and different situations that you might be, don't worry, I will not be talking about the movie, but I think that I want to relate that to something that you see in this very nice, I think, sketch that shows our carbonate wall and the interaction with microbes. And if you look in detail to the boundary in between these two things, you see that there are some areas in which our microbes are building the wall. There is some others in which so they are the good ones. There are some others where they are sort of destroying what it has been built, and they are the bad. And then there are some others in which it's very difficult to say whether they are bad or good. And this is the ugly for us as geologists, because it's very difficult to figure out what is going on. So all this is related to also something that that is sort of summarized in this very nice diagram that I modified from Trumpet from the early 80s, where he shows that there was an interaction in between biotic and abiotic signatures in microbialites, that they were mostly in the macro scale influenced by the environment, while where we were going more to the micro scale, they were influenced mostly by the biology. Of course, further work have shown that this is not exactly always the same, that can change, but it still shows the complexity of this kind of interaction between the biotic and abiotic processes. And here you can see this in this diagram, in this photograph that I have modified from Clément Pollier, that shows that the mineral component, that is what we as geologists, we find, there is an interaction with the biological component, with the environmental component, but what is exactly this interaction, and what is the process that trigger the final mineral components, that is what we see afterwards in nature. So what I would like to do today, and the goals of this presentation, is to show the work that we have been carrying out in the last years, investigating the relative role of these biotic and abiotic processes using living microbialites in modern lacustrine systems. And the final goal as geologists for us is to identify there, if any, distinctive signatures that we can apply afterwards to Earth and planetary fossil record. Since for a question of time, most of the presentation today will be concentrated in the third question, that is the investigation of the relative role of these biotic and abiotic processes in living microbialites. As you can see, is a very vast field in which there are many different aspects to be studied that cannot be done by only one person. So I am showing here and thanking already to my collaborators, people that have been part of our team or that are presently part of the research group, that they go from geomorphologists to hydrogeologists, biologists, geologists, of course, and people working in all what is genomics. So thanks to all of them because all this complexity can only be study if we have a group of people with different expertise. So as I wrote in my abstract, I will show you examples from the Alps to the Andes, and I will start showing you the one that is closer to my lab in Geneva, in Switzerland, that is Lake Geneva, or Lake Le Mans in French. And I will talk to you about some experimental work that we have done in this freshwater lake. This is actually not something new. Already at the end of the 19th century, this guy that we see in the photograph, that it was Francois Alfonso, that was actually a medical doctor, but he was a very interested, he was a naturalist, and I have translated what he wrote here. Is a free English translation from French done by an Argentinian, so I hope it's okay. But this is, he wrote that researchers from Neuchâtel, that is another city in Switzerland, have studied the sculpted, wonderful, developed in the surface of pebbles in the shores of in the lake shores, by the foot of the mountains. And he said that these had been actually formed by cyanophysiid algae. In this, I have to clarify it here that at the time they thought that what is known today as Cyanobacteria, they thought that they were algae, but they were actually referring to Cyanobacteria. And if in this satellite image of the city of Geneva, you can see here the city of Geneva. Our lab is a little bit outside of the image, but it would be somewhere in here. And in this part of the lake, it has been described already back at the end of the last century, in the end of the 20th century. Stéphanie Girard-Clos and some other colleagues work to describe this oidal sand bank here around in this part of the lake, that with a very strange shape, where we have these beautiful ooids that and ovoidal sands. Let me just remind you that this is the way in which these ooids look in Lake Geneva. They have a nucleus of mostly of silica and quartz, and they have alternating low magnesium, calcite, and organic matter concentric layers, and they form in shallow waters. This is about, let's say, an average of 3 meters water depth, or the deepest area is 3 meters water depth where we have the bank. When I remember in most of our sedimentological courses, we learned that these ooids can be formed chemically by supersaturation, physically by accretion, and biologically either direct or indirect. So we know that in Lake Geneva, the chemistry of the water is quite well known, and we know that there is not supersaturation. So we don't consider that, but then we will consider the other two aspects. And based on this, we knew the sand, the ovoidal sand, was well described, but we designed a project that was a PhD project of Karine Play, that where we set up, where here you see the red arrow, we set up this very simple device that was basically a pole with two arms, where we located different kind of like thin sections, thin glasses like this, the size of a thin section, that we previously frosted in order to have a very irregular surface, because microbes like irregular surface, like our teeth, for instance. And then we set up this in this part of the lake. And here we see Karine that was diving every month for 2 years in order to recover one of these glasses to study them in the lab. That, as you have seen in the picture, is very close to the lake, so it was very easy to study immediately. So she would bring these glasses to the lab, and here you can see this under natural light. And this under autofluorescence that help us to discriminate these photosynthetic organisms that we see in pink, and then in white, this bluish white, we see the carbonate. We analyzed that, and then we first of all we found out that there were several organisms there. In this sort of biofilm population there, we have diatoms, we have cyanobacteria, sort of these that are actually these ones. These are filamentous cyanobacteria, and these are coccoid cyanobacteria. There are also green algae and some heterotrophic bacteria like sulfate reducers. So, by the way, heterotrophic, for those of you that are not very familiar with the terms, heterotrophic means that they need another kind of nutrients, and they are not photosynthetic like the green algae or the cyanobacteria. So if we go back to our experiment and we look in detail at the low magnesium calcite and biofilms. Here, this is the aggregate. Look a little bit ugly at this scale, but if we look in detail, what we find there is this beautiful sort of spaghetti-like, to be like with the title of the talk, that are the filaments of the cyanobacteria, that are also some coccoid cyanobacteria, and some crystals of low magnesium calcite. All these are sort of enclosed in EPS, that is the extracellular polymeric substance, that you will see this name in several parts of the talk, because this extracellular polymeric substance is nothing else than this sort of material, a slippery material that you find in every place that you have microbes, because they like to live in this sort of matrix until they form a biofilm. So when we analyze the surface of a sand grain, now sort of a fossilized ooids, if you wish, you find, excuse me, these remains of cyanobacteria filament that are very similar or identical indeed to what we see in the in situ slide. So the experiment, what we were trying to do, that was to reproduce what was happening in the grain of the ooids. We managed to do it in this experimental place on site. So, but the nice thing of this experiment, is that then you can pick them, the living microbes from your slide, and you can grow them in the lab. And we have done these liquid and solid cultures. And why do we do that? Because if we have the organisms, if we identify them, we can change the physicochemical parameters and run different experiments in the lab. Then we can model biochemical interactions, and then we can constrain the biological role. That is what we wanted to do. So this is what we have done, and this is the microbial culture approach. I will not go much in detail, but I will just simplify the thing for you. That is, we did experiment with light and without light, using only a media that is called lubricant broth, that is quite common in microbiology, to grow heterotrophic microorganisms. And what we see here is that only with light, our photosynthetic microorganisms would precipitate, would form, first of all, the EPS, as you saw in the previous one. We would have biofilms, and we would precipitate low magnesium calcite of the exact same composition that we had in nature. So, you know, this helped us to clearly know that it was necessary to have cyanobacteria to precipitate this kind, excuse me, this kind of low magnesium carbonate. So, and we analyze some of these samples under confocal microscopy, that where we can dye the different components of the samples. And this is two dyes, this is with 3 dyes, and what you see there in blue is our low magnesium calcite, and then you see in green the cyanobacteria and some heterotrophic bacteria, but they seem to be a little bit on the side. And then we have the EPS in gray. So what we see here also, this confirms that our mineral phase is really involved in this EPS. And we can even see better in this TEM image, where you see the mineral phase completely surrounded, or I would say enclosed by this EPS, where this is an sort of EDX of this part of the sample to show that this calcium is coming from this carbonate phase, low magnesium carbonate. So with this, we still wanted to do something that now is quite common in this kind of study, that is to use a molecular approach and to identify whether our experiment was really doing the same thing, and that we were not changing so many conditions in the lab. We analyzed the ooids, the biofilm that were harvested in the in situ experiment, and the enriched cultures that we did in the lab. And we use an approach that unfortunately we are very much used to here these days, that is a PCR. We did a PCR of the extracted DNA on these organisms, on these different samples. And what you are seeing here are the biofilm slides in the natural experiment, the ooids, and the enriched cultures. And what you see there that I'm highlighting here is that in general, the distribution of these bands that actually correspond to these different genes, they are the same. And they, of course, as you would expect in the ooids, you have a lot less because most of the organic matter had been partially destroyed because the organisms are dead, different with the enriched culture where you enrich some of them a lot more than in the natural system. So this model that we published in 2008 actually has been also applied to another places, and for instance the Bahamas, and I'm showing you here this diagram because I think that is a very nice sketch of what is happening after a review by Diaz and Everly from 2019, where you see that in the Bahamas, and it's the same like Geneva, in the bottom, you have kind of like an EPS colonization where on a sleeping stage in the seabed. And then you have the microbes that like to live in this place, in this EPS, and they develop and they start precipitating, forming, for instance, in this case, is mostly amorphous carbonate. And then they, with the turbulence, they go to the suspension stage and they are start to aggregate this amorphous carbonate and they start precipitating. And well, this is a different circle, but it's that in Lake Geneva, because the water, the composition of the water, marine water is very different. But what we want to show here is that these guys are the good ones here because they are in this part of our world in which they are building up our carbonate wall. So these are the good ones that they, and it's in this case easy to identify them. But now I will bring you to the Andes, and I will show you two examples of similar studies that we did in two basins. One is the Maquinchao Basin in northern Patagonia in the Argentinian side, and the other one is Laguna de los Cisnes, that is in Tierra del Fuego in the Chilean side of Patagonia, and the closest, one of the closest sites to Antarctica. So let's move first to the Maquinchao Basin. The Maquinchao Basin is very interesting because it has living and fossil microbialites. This basin had been studied for many years now for different groups, because as you can see here, there are a beautiful a shoreline that had been associated to the times in which the lake was a lot larger than today. The two lakes were only one. And what it was interested for us, we were interested in, is that all that you see here, I hope you see these little points in red, are outcrops of microbialites. And what you see here in the green, with the green arrow, is places where today we have living microbialites. So again, we have living and fossil examples of the same microbialites. So during the thesis of Ines Emar in our group, she looked at this in detail, and she identified 6 different kind of microbialites that you can see here. I will not go much in detail on this because as I said at the beginning, I want to concentrate mostly in the microbial, in the living ones. But if you are interested, you can check it in our IAS open access journal, the depositional record, and where you can find all these, a paper where all this description, and even this part of the presentation where actually we concluded that the occurrence and the distribution of these morphologies, or the different types of morphologies that you excuse me, you see here some some photograph. They depend on the type and size of substrate, on the groundwater input, the topography, and on the lake level changes. So in a way, you see that all these things correspond to what we would say the environmental influence. These different environmental aspects were sort of ruling the fact that we would have this kind of big microbialites, or this sort of extended one, or the little one that you can see here. We managed to date some of these microbialites that you can see here with the red arrow. We noticed that as expected in this area of the world, during the last glaciation, the lake was a lot higher than today. And during the deglaciation, it was going down. Here you can see in this diagram, the elevation in meters above sea level and the age. So what we are showing here is the last deglaciation. And notice that the elevation is not on scale. And this is, for reference, the level of the Karilavken Grande, this lake, in 2011. So you see that we only have microbialites when the lake was a lot higher than today, and therefore with a completely different chemistry than the one that we have in these two systems today. But as I say, I want to show you mostly what we have done in the living one. The living ones are something, this is one, a small one. They are a lot smaller than the fossil ones, and they are located mostly in the river that join both lakes today. And if we cut it here, we find that we have a very clear green biofilm in the uppermost part. This is in the millimeter scale, and a beige white biofilm in the lowermost part. And when we look at them in the microscope, we see in the green that the green biofilm is mostly composed of filamentous cyanobacteria, again our spaghetti here, and you see that they are living because this is with epifluorescence. The microscope, you see the color here that are the living filamentous bacteria. And when we go to the beige or white biofilm, in the lowermost part, we see that it's mostly formed by translucid filamentous cyanobacteria. There are also some coccoid cyanobacteria, but we have a lot more mineral phases, that is, again, low magnesium calcite. And I will walk you through these images that go, ACN picture images that go from a larger to a small to a larger scale in this sense. And this is for the green biofilm. And what you see here is that we have diatoms, broken diatoms. And when if we sort of zoom up in this green square, we see that there is a lot of EPS diatoms that are partially dissolved. And then if we go more in detail, again, we find crystals, or starting, let's say, crystals of low magnesium calcite. And if we analyze this more in detail, we see that we have sort of mineralized, we call it nanoglobules, and a nanoporosity that start to develop and sort of alveolar network here. You see here that they are sort of joining together and they are starting to form these crystals that you will see in the next slide. Just to tell you that in our EDAX analysis, you find the calcium and you have a little bit of magnesium and here the oxygen and carbon. All this is it shows clearly that it's carbonate that has been also analyzed independently. Using another methods. Now, if we go to the beige white biofilm, what we find also, again, we have somehow a lot more EPS. And what is nice is that you see that our crystals are a lot much developed. We have these sort of platelets of EPS. We have is here. They are very well developed. And these crystals are always embedded in an EPS. So this is, at the end, you arrive to get this sort of ehedral rhombohedral that is typically a very well formed low magnesium calcite crystal. So this is also, there is an early paper from our group where you can find more details about this. And something that it was really interesting and that we have also heard in another presentations in this series of webinars by Maurice Tucker, where he was talking about virus, because we found very interesting, and I tell you, this was a lot before COVID, but we found elements like these thylakoid membranes that are typical of virus in the green film, biofilm, but a lot more in the beige white biofilm. So they are, we can see in this beige white biofilm the early state of degradation, as you can see here, and then you can see in another places a very advanced state of degradation with forms that are typical of virus. The role of virus is still not very well understood, and their role in mineralization is even more difficult to explain. But some people claim that they are associated to the formation of the carbonate, while some others say that they are actually degrading the organic matter and they are not that important in the precipitation of the mineral phase. In any case, what is important also too, that maybe I forgot saying here, that is in the beige white biofilm, we have a lot more sulfate reducing bacteria. So the green biofilm is mostly formed by cyanobacteria, while this one are formed by mostly by this virus, degraded cyanobacteria and also sulfate reducing bacteria. So if I have to summarize, and notice that I wrote here microbialites from 2011. I will explain you this later, why I wrote that. Because at this time, we got that the presence of microbes in both green and beige layers they are always there, but they are in different proportions. That is important, in different abundance. There is an increase of dead cells of cyanobacteria, as well as some other heterotrophs in the beige layer, and they are apparently the bad. These are the ones that are destroying precipitated, but at the same time destroying some of the things that are formed. And then we have this virus, as I say, that we don't really know what they are doing. As I say, people say that they are the bad. Some others say that they are the good, but in any case, they are the ugly because we don't know exactly what they are doing. There is something that I didn't have time to talk about it, but that is very interesting. That is that these EPS are enriched in a phase that is silicium, calcium, and magnesium. Remember that I mentioned that there were pieces of diatoms that are broken. And in the same case that in the ooids also, that there were some pieces of diatoms stick to the experiment that we did in the lake. It's interesting because apparently they are mineralized at the beginning as amorphous magnesium silicate phases that then will become this low magnesium calcite. So they are the good one, this part of the EPS that contain. So one thing that is also important is that the carbonate crystal in both the green and the beige white lamina, they have the same composition, but they have different morphologies in both biofilm. So then, as I say, we went many times to the field to do these experimental studies and to do a deep hydrogeological study too. But we noticed when we went in 2015 that the situation in the living microbialite was completely different. We didn't know exactly why, but we have some clues in the sense that we know that at the end, the first campaign was in 2011, and 8 months later, there was a major volcanic eruption in the Andes that covered completely the area, in some places with more than a meter of volcanic ashes. And we obviously it had an impact in all living things, including microbes. But they, of course, came back. Look at this photograph. This was in some living ones that we sampled and we analyzed, and we did some, we were, since we knew a lot more than when we went in 2011, we thought that we wanted to do more sort of genomic study, that now they are a lot more accessible and easy, and we work with biologists and microbiologists to better understand that. And we did this analysis that you see here, and for our surprise, this is different samples of these living microbialites that we sample at sort of a millimeter scale. That what it means top, center, and bottom in 3 different sites. And what you see as different colors is the amplicon sequence variant. That, don't worry, this is something like it's telling you about the diversity of the different film that you had present in the sample. And for our surprise, there were almost no cyanobacteria. Remember that I told you that the uppermost part there was formed by cyanobacteria, but there were mostly groups of sulfate-reducing bacteria. And all this, I will not go in detail to that, but I just want to point out that in almost 4 years, there was a major change in the diversity of these microbes. And indeed, inside this group of the Sulfobacterota, there were of this class, there were several groups that we could identify. We have recently published this at the very end of the 2021 in the Latin American Journal of Sedimentology and Basin Analysis. That is also an open access journal. So if you are interested in learn more about this, I invite you to check it out. Now I will like to say that in this example, we find that we have everything, because we have the good one that precipitate, the apparently bad one that also dissolve or destroyed part of this carbonate early form, and they're ugly because, for instance, we have the virus that we don't know exactly what is happening, and they are covering and maybe going even at some moment to sort of an abiotic phase that is very difficult to distinguish. And then I will move to the very last example. I hope that you manage to see this nice video that I made in Tierra del Fuego just a few days before the lockdown of COVID. We came back to the field with Clément Pollier in February 2020, 2019, sorry. And this place is wonderful because it also contains fossil and living microbialites. And we did, this is part of the master thesis of Clément Pollier, the University of Geneva, that he just moved a few days ago to Erasmus in Miami, where he will be working on his PhD thesis. And we identified kind of like in the same case that in the previous case, different shorelines where we have, you can see here a shoreline at 13 meters, 11 meters, and 7 meters. And you see that it's plenty of microbialites that are shown like that. So, and the nice thing is that we have the present shoreline, we have the living microbialites, and next to them, the fossil ones. So Clément identified 4 different types, and spherical, elongate, crater-like, and this columnar one. And he relayed mostly the position of these different types to the energy and to the accommodation space. So environmental factors, but also, as you will see, to some biological ones. And we have these morphologies that are distributed differently along outside the lake, around the lake, and in the lake, the living ones. So I will show you one of these tall morphologies. If we make a cut in this sense, we see that we have these beautiful sort of tubes like that look again kind of like spaghetti, thick spaghetti. And then if we cut them in this sense, we see here that these tubes, the different size and distribution, that are the ones that make sort of like the structure of this different microbialite. And Clément identified that actually they correspond to a green algae that is called Percursaria percussa, and that are building this kind of structure. But there are not only these, but also there are a lot of microbes involved in all that. Let's have a look in detail to this, a piece of this tube. And what you see here is that there is kind of like almost an ooid distribution of with different composition. This is a petrographic microscopic view under normal light. This is under cross Nichols, and this is under cathodoluminescence. And what you see here is that they are different, very different mineral composition. And this is what allows us to do an interpreted view of these different compositions. I take advantage of this presentation to show you something new, a method that we are using in Geneva that is called Chemscan, that combines the mineralogy, the density, and the textural data, including porosity and pore morphology. So we use this to generate sort of vertical logs if you're looking to a core, before, but also to support sedimentological studies that provide indication of variations, for instance, in grain size, and also this and different aspects that will help us to reconstruct the positional environments. But what we were mostly interested in this case, it was to generate true mineralogical logs with quantification of all mineral phases. So, and this is what we did in this same sample you can see here. And what we identified is that these different kind of compositions are monohydrocalcite in the external section and is dolomite inside the central part of this. When Clement first showed me this, I said, well, are you sure that it's dolomite? We have to look more in detail to this. And we did a lot more analysis using Raman, a new technique developed with Raman spectrometry. And we also did XRD, etc. So we are sure that this is dolomite, but I will not have much more time today because I see that it's also almost 50 minutes that I'm talking. Sorry about that. So I will move to something else and finish the talk in a few minutes. But if you are interested, hopefully all this information will be soon published. And I hope you will look at the papers if you're interested in this. And we also did some analysis using genomics to see what kind of organisms we have there, and we are working. We have now, this data is relatively new, and we are now working on this. But you can see here that we have cyanobacteria. We have different kind of organisms that we will try to relate this with the model of formation of these microbialites. So I hope I also show you that here in this example, again, we have the good, the bad, and maybe the ugly, although we need to sort it out better, and we are working on that right now. This, as I said, this is a very new data set. And with this kind of like, I would like to finish, and I hope that I managed to pass the message that although we have the good, the bad, and the ugly, more and more we have techniques and approaches to better understand and try to separate them and their role in the formation of our carbonates. I think that this also relates to one of the great debates that we have seen sets online about whether the present is good to understand the past, but I think that probably as it came out on this great debate is what we have, and it's probably the best way to sort of try to understand better this kind of different processes. And just to leave you with a little thing to think about it too, I would like to show that all these studies are also extremely important for astrobiological studies. And this is a selfie of Curiosity rover in Mars that as shared with our group, the Curiosity. We are very curious about to see what can we learn in our Earth, outcrops and living microbialites, to sort of see if we can see something in another planet. And I just would like to leave you with this picture that is a comparison of the Laguna de los Cisnes, the last example that I show you, the structures and spherical and flare morphotypes that we found there, that looks really very similar to what we see in these images provided by by the recent and the ongoing study by curiosity in Mars. So perhaps if we learn a lot more about this and we can compare this with the fossil record, we can use that to better understand what is happening first of all on Earth, and what had happened in the past on Earth, but also what maybe had happened in another planet. And before finishing, I would like to thank very much all our funding agencies, in particular the Swiss National Science Foundation, that had been extremely helpful funding us during all this year, as well as some other foundations from Switzerland and from Argentina. And with this, I would like to thank you very much for your attention and patience after these almost 55 minutes. And I will be open to any question. Thank you.
Thank you so much for that really great presentation. So anybody that has any questions, I see we already have one popping up in the chat. So you can please send your questions to everybody, not just to SEDS Online or to myself. Make sure that everybody can read them, and then I will read them out. Okay, we have our first question. So I guess I'll be getting, we have two already. So I'll be getting to mine later on, hopefully. But we'll start with Morris Tucker's question. Lovely images, Danielle. Good to see your viruses. Were they being mineralized? And did you find any vaterite basin case?
Sorry, can you repeat it? I didn't.
Yeah, there's a couple questions. There's a couple questions, so I'll break it down. So he wants to know, first of all, if the viruses are being mineralized.
Yeah, well, it appears in some of the images that we have that they are mineralizing. And some of I think in one of the images I show this sort of nanoporosity that it could be some of the virus that are mineralizing, but I take with caution right now because the images are not so clear.
Okay. And then he also wants to know if you found any vaterite.
No, we didn't, but we didn't look much for it either. So yeah, no, at this stage, no.
Okay. And yes, Morris is coming from Bath. Our next question is from Roman. It says, hi, Danielle, very nice presentation. Thanks a lot. Regarding the lake, I can't pronounce it, sorry, great, but Mackenschau, yeah, okay. Regarding Lake Mackenschau, I was wondering why the living microbialites are almost exclusively found in the river that feeds the lake, while the fossil ones look like they're only found on the paleo shoreline.
Yeah, thanks, Roman. That's a very good question. And I think that this is one of the evidence that shows that in the past, the chemistry of the lake was very different to what is right now. Right now, they don't allow to leave any kind of, I mean, probably there are some microbes, but there are not this microbialite formation because the composition. While in the river, because there is a lot more groundwater coming. I didn't talk about this, but we did a detailed hydrogeological study that shows that the chemistry of the river is the one that allows these microbes to develop in there.
Okay, and once it gets to the actual lake, is it too diluted then, the ions, or is it
yes,
just not
they are. It's a completely different composition because most of the lakes today, they are fed by groundwater. But while this groundwater arrive, since it's a very dry, I didn't say that, but it's 200 millimeters per year max of precipitation, so it's quite dry. And then when the water arrive already to both lakes, there is a lot of evaporation because there is a lot of wind too. We are talking about 100 and 150 kilometers per hour. I mean, sometimes it's impossible to do field work because the wind, what it also trigger a lot of evaporation and concentration of elements in both lakes.
Yeah. Okay, we'll move on to the next question. It's from Peter. He says, thanks for an excellent talk, Daniel. I'm curious about the dolomite you found in the middle of all those tall microbiolites. What was the calcium magnesium levels and the cation ordering? Do you
Yeah.
think, sorry, a little bit more. Do you think the present has anything to do with magnesium-rich EPS that you mentioned?
Yes, I do think that, I mean, as I say, Clement is working on the papers, but what we notice is that there is a lot of magnesium in this EPS, and there is also this sort of silica, magnesium, calcium composition, the same that in or compound, the same that we have seen in Makinchao. But we are working on that data, and hopefully a model, we will provide a model. But we were quite surprised of having this dolomite in there. So.
And then I guess he also was questioning about the ordering of the dolomite. Is it
Yeah, it's quite well ordered. Yeah, quite well ordered. It's not perfect dolomite, but it's quite well ordered. And as I said, we did a nice study with the Raman that is really showing peaks that, of course, that are not perfect because it's not perfect dolomite, but they are quite well developed and easy to differentiate the different things. The different minerals.
Great. Our next question comes from Ross Campbell from Queen's University in Canada. Thank you for the fascinating presentation. What do you think the relationship is between the sulfate reducing bacteria and the carbonate mineralization, the increased crystallinity, and also were any clays present?
Well, in several of the well-known models, for instance, the one from the Bahamas that shows that these sulfate reducers are important sort of in helping to precipitate the carbonate. So it appears that while the photosynthesis is important in the uppermost part, maybe changing sort of the microenvironment to precipitate the carbonate. In the lower part where you have these mostly heterotrophic microbes, they use during their metabolism, to survive, they change the environment, microenvironment, and they precipitate the calcium carbonate. That's the result of their metabolism somehow. Yeah.
And he wants to know also if there were any clays present.
Any?
Clays?
Of course, yes, there are. And particularly in the last example, in both. And we are working on that too. There are clays, yes, certainly.
Okay, our next question is from Saeed, and splendid presentation. I would like to know the reason for low mag calcite in the lake, because in sea rifting influences sea chemistry for the lake is unknown.
Well, I would like to know too, but because, you know, we, as I said, this is very recent, but there are 3 lakes in the area very close, and only this lake has microbialites, and the other 2 don't. And they are at the sea level, and maybe it's because in the past this lake was mostly more sort of joining the ocean. I really don't know. We have the chemistry of the lakes that are quite different, of the 3 lakes. This lake also in the last year in the last, let's say, 30 years, had sort of slowed down quite a lot because human impact, right? But the microbialites are a lot older. They are even from the early Holocene. So it's not only human impact on this. But yes, the chemistry has to do, and we need to go more in detail on the different organisms and see what they are doing and why this is happening, this sort of concentric distribution of minerals. hopefully this will be published soon.
Okay, yeah, so everybody keep your eye out for these new publications. Okay, so anybody else have any more questions, please type them into the chat, and remember to let us know where you're watching from so that we can, yeah, just sort of share the information. Okay, we have one more. Hi, Danielle, very informative presentation. You showed one slide about PCR analysis and extracted DNA. Could you try to explain, so what do you try to explain there? He wants to know what the PCR data is trying to tell us.
Yeah, well, in the first example, in the Magnesium basin, the well, let's say in the ooids, what I was trying to show is that the distribution of the same organism, is the same in the real sample, in the ooid, in the experiment, and in the lab experiment. And the reason why we did that is because quite often when you do this kind of jump from the natural system to the lab, people claim, oh, you know, you just maybe grew up a different microbe. So that's why we wanted to show in this case that the experiment in the lab was compatible and clearly the same thing that we were seeing in the natural system. That is one. Then in the second case, when we did also PCR, and well, actually we did more sophisticated methods in 2015, it was just to show that the distribution of what we were expecting to find a lot of cyanobacteria and things that we didn't. Maybe it's a problem of the analysis itself. We are looking into that too, if we use different kind of like reference material. But the idea is always to combine what you see in the microscope, what you see in the genomics. Because once that you identify what is in the genomics, is a lot easy to understand. Looking at their ecology, their microbial ecology, you can maybe better understand the process behind the formation or what they are doing there. Absolutely. I hope I answer the question.
Yeah, I think so. It's pretty informative. Okay, so in case anybody has any more questions, please type them into the chat. While we are waiting, I had a couple of questions, so maybe I can get to mine. You showed one TEM image from the ooids from Lake Geneva, and I'm just curious, it was a very sort of complex crystal structure. And is that sort of a preliminary stage, or is that the final crystal structure that you see?
No, it was a preliminary stage. And it was a preliminary stage because this picture was, this image was from the experiment. So in the lake, so, you know, it was just starting, let's say, starting to form the crystal.
So once those crystals sort of are removed from the EPS, or the EPS is removed from around the crystals, let's say, does that really complex texture sort of start to fill in? Or what is the...
Yeah, in general, is more filling and more better develop the structure, you know, I think.
Yeah. Okay. Yeah, we see some similar things in the freshwater system in the Everglades, sort of that very complex, like trellis-like structure. So I was just interested to see that. What do you think that means about the fidelity of the actual, say, isotopic data that we take from the carbonates? If their initial structure is then being sort of even very early stage, but later filled in to a different...
it's a very good question. We tried to do that, but unfortunately, in the case of the Maquinchao, that we did a lot of isotopic data, it was very difficult to sample with the resolution that you need to really be sure that there is biological impact or not, because actually the samples that the isotopic values that we had, they were mostly a combination of, for instance, the laminated area and the other one. So the sampling wasn't with a good resolution to do it. That in the ooids, on the other hand, we did nanosims. And there is a paper in geology by Pacton et al. That I put the reference, I think, where we could identify these different isotopic composition. distribution of, as you say, the biological impact, you could clearly see it in there, in the isotopic values of the UITs. But
And
in
what
the other,
isotope system did you measure? Was it oxygen or?
oxygen and carbon, yes,
Carbon?
both.
Okay,
Yeah.
very cool. I'll have to go back to that reference. Okay, one more question from the audience. Thank you very much for this interesting presentation. I'd like to understand how in such cold conditions as during the LGM, the bioconstruction was maintained. How can we explain the high levels of the lake during this period in Geneva?
Well, there are two questions there. The first one is, if you think about it, in the second example that I show you is southernmost Patagonia, so it's extremely cold. And actually the local people told us that in winter, these shores of the lake where there are most concentrated the living microbialite, is frozen. So, you know, microbes can live everywhere. If you think just simply in Antarctica, we have beautiful examples of underwater microbes, under very cold conditions growing there. So I don't think that this is an issue, the temperature. Although it's true that we are mostly used to think in microbes and stromatolites, for instance, in the Bahamas or in Shark Bay, but we have seen in both cases in Maquinchao and in Laguna de los Cisnes in Chile, both cases are very cold in winter, and you have microbialites growing like very happy there. And then for the second part of the question, that is the fact that we have high level. How do you explain? Well, all this area, if you think from Lake Titicaca, that is about at 15 degrees south, and you move to about 41 degrees south, that is the position of this lake, all the lakes are very high during the late glacial, because all this belt of humidity was very important there, and there were a lot more precipitation than in the late glacial and then during the Holocene. While if you go to the south and you go to you go to, for instance, Lake Cardiel and in other sites, they are dry during this same period. So there is a gradient of moisture that is relatively well known in Patagonia, and even starting from Bolivia and in Lake Titicaca. So it's something that is quite well known, that you go from high lake levels in the full glacial to in the north, and to dry conditions in the south.
Okay.
Yeah.
He says thank you. Yes, Khalil coming from Morocco. Okay, I had one more question maybe we can get to before we let everybody go. I was interested in the Chemscan. I was just curious. I mean, it looks like very interesting data sets that you're able to obtain. What's the actual resolution of some of that data?
Yeah, well, it's a very, very high resolution. You can go, I don't want to say a mistake, but I think you go down to something like 5 microns, you know, and you can really obtain, depending on the database that you have in your machine, you can identify, basically the machine will identify the mineral and will map it as, you know, in the same way that you do with microfluorescence, that you can do a mapping of, for instance, something like these UITs very much in detail. You can do it also with the Chemscan, but with a better resolution, a lot better resolution. But of course, it's a lot longer time of analysis and a lot more expensive, obviously. So, you know.
On the period of what, like 1 day, or is it hours?
But depends the size of the sample. The advantage is that you can put many slides or many thin sections, and the machine will run it maybe for 1 day, 2 days, or something
Mhm.
like that, depend on what the area that you want to analyze. And that. It's the first time that we are using these in lake systems, as far as I know. But basically, you can prepare thin sections of any kind of microbialite, because all these examples that I show you, they were very, very sort of non-solid samples. So we have to sort of solidify them with araldite, and then we make thin sections, and then we put it in the machine. And it works well. I think there is a lot of future to do different things on this.
Yeah, it looks very interesting. Okay, well, I think that we hit all of the questions. Daniel, thank you again for this wonderful presentation. We're very happy to have had you today. Thank you everybody in the audience for coming as well. And don't forget to join us next week when we're going to have a seminar by Ricardo Gatter on submarine landslides, and we will see you then.